Inflammation, color, duller, rubor, what else? There is one more, pallor, very good. Pallor is there? Pallor is not a part, I believe. Rubor, duller, color, tumor, very good, very good. Now, what are the vascular changes in acute inflammation? All of them we find, but hydrostatic pressure is increased, and that is responsible for the edema which you see in the case of the inflammation. Degeneration of the basement membrane is basically mediated in inflammation by which important subset of enzymes? It is the metalloproteinases which will cause the degeneration of the basement membrane in inflammation. Now in the basement membrane, who is the predominant glycoprotein that we have, and it is the destruction of that responsible for the basement membrane degradation in case of inflammation. It is the laminin, which is the most abundant glycoprotein which you find in the basement membrane, is what need to be remembered. Now there are some small proteins, connexons, annexins. You must know 1-2 words about them. So connexin is the protein which is involved in intercellular connection. So what are connexons? Connexons are the complex protein assemblies. They traverse to the lipid bilayer in the plasma membrane. They form a continuous channel, and the pair of connexons from two adjacent cells join to form a gap junction. Just like the buttons we put for the shirt, the role of the button is played in the shirt by the connexons, if it is a lipid bilayer, is what need to be remembered. Then what are adhesins? Adhesins are the microbial surface antigens that typically occur in filamentous projections like pili and fimbriae, and they bind to specific receptors on the epithelial cell membranes. Then what are integrins? The third group of proteins when we talk about cell injury and inflammation. Basically they are a family of glycoproteins. They serve as a extracellular matrix glycoprotein receptors, and they are involved in the cell adhesion, and they mediate the adhesion of the neutrophils to the endothelium. So that is where the integrins come into picture. Now let us go to the next question. Where do we find laminin? As already we discussed, it is a basement membrane where we find the laminin, and that forms a very integral component. Now what is special about natural killer cells and cytotoxic T cells? The function of both of them is they both are antiviral in nature. Now let us quickly talk about natural killer cells. If a T cell, cytotoxic T cell, wanted to destroy a particular cell, what is required, doctor? It requires a warrant. First of all, that yes, he is a murderer, so I can kill him. So who gives that warrant to kill the given cell? First it should recognize it is self or non-self, hamara ya paraya. So it is the expression of MHC antigen which is very important. Looking at MHC, a cytotoxic T cell will recognize it is a foreign or a self and destroys it. So the destruction which is done by the T cell is MHC restricted, is what need to be remembered. But what is speciality of NK cells? It is not MHC restricted. It does not require MHC compatibility, expression, recognition, and then destruction. It is more or less like a communist government. Take to the outskirts of city and shoot. Done. There is no trial or anything. It's like a war, the war hanging which is being done. So NK cells are first line of defense. Naturally, no. If you have to make virus to attend a court, go to appeal and do all these things, there will be a lot of time. So you should destroy it. You suspected that it's a virus infected cell, destroy it without much of appeal or trial or a court or anything. So NK cells will do that. They are the first line of defense. MHC unrestricted kill is what they do. Cytotoxic T cells, they will basically cause a class 1 MHC restricted T cells, and their destruction of cells is MHC restricted. Now in the intraepithelial area of our mucosa we have a lymphoid tissue called mucosa associated lymphoid tissue, MALT. So that has got a lot of T cells is what need to be remembered. Now in the inflammation, the next question of all India, inflammation is the second chapter of Robin's pathology, which we have to quickly review before going to exam. Macrophages basically derive from monocytes, as all of you know very well. Now one more question on NK cells. They are the large granular lymphocytes basically, and they comprise about 5% of the total lymphocyte count, and they express the immunoglobulin G Fc receptor, and they are not MHC restricted is what need to be remembered. Now let us see how does an NK cell basically destroy a virus infected cell. Is this illustration given to you? It's not there? Okay. What we'll do is actually all this content is prepared in the last moment, overnight. So last week and every week illustrations we give in a separate set of papers. So it become easy for you to use them. So they are not there in the flow of text. So if you take NK cells, there are certain granules which are capable of causing the cytolysis, and they come to a healthy cell, but a healthy cell will have a very good expression of MHC class 1. So such a cell it won't do anything, but a virus infected cell, the MHC class 1 is not expressed. So is MHC class 1 is there or not? But what type of MHC it is it self or not self, all that discretion NK cell cannot read. Only thing it will check, passport hai ya nahi hai, nahi hai to chalo andar. So that is what it will do. It will not check which country passport, when is valid up to, that is all done by cytotoxic T cell. So if there is a low level or no level of MHC class 1 expression on a virus infected cell, then that becomes a target for the NK cell and it will go and destroy it, is what you need to fundamentally understand. So this is an intracellular pathogen, it has infected the cell. The moment it has infected, then there are two situations. Number one is the MHC class 1 downregulation can be done by the virus, and if it is downregulated, NK cell will destroy it. If it got upregulated, then cytotoxic T cell will kill it. So finally cytotoxic T cell and NK cell together, like Ram Lakshman saving the Yajna, they will basically save our body from being destroyed by virus cells, is what need to be remembered. NK cells are the large granular lymphocytes, morphologically larger than T and B cells. They contain what are classically called eosinophilic granules, is the hallmark of the NK cell. And there are three important receptors, doctor. This cluster differentiation, CD receptors, you must be very precise, numerology number game. CD16, CD56, CD8 and CD2, all of them basically are expressed by the NK cell. Out of them, CD16 is the surface receptor for the Fc portion of the immunoglobulin G, is what you need to fundamentally remember. And some NK cells will also express CD3, and they are called as NK T cells, and they become activated by interleukin 2, and they are also called lymphocyte activated killer cells, is a name given for this special type of NK cells which show CD3 positivity and which are basically stimulated by the interleukin 2, is what need to be remembered. So there are two important features about NK cells. Typically it does not involve any antigen antibody reaction, and it leads to a direct cell lysis. They are capable by releasing the eosinophilic granules, which are like a nuclear bomb that they are having, is what need to be remembered. Now let us take up the next question. Fibroblasts, from where do they derive from? They derive from the mesenchyme. Then what is the lifespan of the various types of cells, is another favorite question. So granulocytes like neutrophils have 4 to 8 hours, and if they go to tissues they will have a little longer life, 4 to 5 days. And platelet will have 10 days, monocyte will be in about 10 to 20 hours in circulation, is what has to be remembered. Now we come to a very important area. Ilam, Vikam, Ikam, they are the three mantras of inflammation. Vascular cell adhesion molecules, endothelial cell adhesion molecules, Ikam. Now let us talk about them. If there is no LAM, which is the leukocyte adhesion molecule expression, How does they clinically present? Fundamentally, why do we require all these adhesion molecules? Basically, a cell, if it is unattached, it is susceptible to apoptosis. Only when it is attached, then it is protected against the apoptosis. It's like bachelor and married, which is very, very important for the cell. So it is the ECOMs, VCOMs, ELAMs and LAMs. They all basically will decide whether the cell become adherent or non-adherent, and if they are not so, then that lead to susceptibility to infection, decreased lifespan, etc. So it is the delayed closure of the umbilical cord which is the marker of the congenital lack of leukocyte adhesion molecule is what need to be basically remembered. Now, who mediates the interaction between endothelium and leukocyte whenever there is an inflammation? We need to remember that there are four important group of molecules which are important in inflammation: selectins, integrins, and ECOM like molecules, and mucin like glycoproteins. They are all very, very important. Now let us have one comment about each of them. Among selectins, there are P-selectins. They are important for the rolling at the time of inflammation, for the neutrophils, etc., etc. Neutrophils, monocytes, lymphocytes, for them to roll you require P-selectin. Then similarly, E-selectin are very important for rolling adhesion to activated endothelium. ECOM1 is important for adhesion, arrest and transmigration of leukocytes. VCOM1 is important for the eosinophils, monocytes to undergo adhesion, and accordingly there is a role of the various adhesion molecules in inflammation is what need to be remembered. So out of them, ECOM1, VCOM1 and CD31, all of them basically belong to the immunoglobulin group of family. Now, what are defensins? One of the options given was defensins, endothelins are also there. Then what is their role also? Let us look into it. Defensins are the cationic arginine rich granules and peptides found in the eosinophilic granules of neutrophils. And main role of defensins is they have a very broad antimicrobial activity against bacteria, fungi and enveloped viruses. So that is the main role. They are found in the eosinophilic granules. Defensins is what need to be remembered. Then what are endothelins? Endothelins are basically a potent endothelial derived vasoconstrictors which are important for the vasoconstriction which occurs after injury. Now, what is one of the famous theory about pulmonary hypertension? Young female comes with a diffuse thoracic pain, loud P2 is there, parasternal heave is there. You discovered that she is a case of primary pulmonary hypertension. Why called primary? Because she does not have any ventricular septal defect or ASD or PD or any Eisenmenger complex. She does not have any hemoglobinopathy or hypoxic state. In spite of all that, she has hypertension, pulmonary hypertension. In that situation, we call it as primary pulmonary hypertension. One of the present theories about primary pulmonary hypertension, why pulmonary vessels undergo vasoconstriction in them is endothelins are implicated. It is excess release of these endothelins which will induce the pulmonary vasoconstriction leading to pulmonary hypertension. So what is one simple way that you can treat pulmonary hypertension in them? Endothelin receptor antagonist monoclonal antibodies we have discovered to treat pulmonary hypertension. We are trying to antagonize endothelins from going and binding the receptor and causing vasoconstriction. Second is endothelin basically interacts with nitrous oxide, and that is also implicated for the vasoconstrictive ability. So that is the reason we give sildenafil, which is nitrous oxide stimulant to antagonize this endothelin mediated vasoconstriction which we see in pulmonary hypertension is what need to be remembered.